All right, welcome to another week of Soul Seminar Online. My name is Chelsea. Today I'm going to be introducing Andrea Berardi. She completed her PhD in the University of Virginia with Doug Taylor, studying the local adaptation of Selene vulgaris. She did her postdoc with Stacy Smith at CU Boulder, studying the evolution and patterns of flavonoid evolution. She continued on and did a second postdoc with Chris Kameyer at the University of Bern, studying the molecular genetic mechanisms of pollination syndrome evolution in Petunia. She is now currently a research fellow at the Harvard University, working on floral color pollination syndrome evolution and speciation of Selene. So with that, you can share your screen and go ahead and get started.
Great. Thank you, Chelsea. Okay, then I'm going to switch here. Okay, is that still the right right view?
Perfect. Yeah.
Great, thank you. Okay, so thank you for having me, and I really appreciate everybody also starting 15 minutes late for me. So, like Chelsea said, my name is Andrea Berardi, and I am going to talk today about the work that I've done in Petunia on the evolution of floral color. And so I think just right up front, I'm going to start with the acknowledgement. So this is my postdoctoral work with Chris Kuhlemeyer, and all of this work was, it was a super group effort with members of Chris's lab, as well as collaboration with Loretta, as well as Aureliano. And it was just a huge group effort. So I'd like to thank lab members as well as our funding sources. Okay, so many phenotypes, as we know, especially floral color, are the products of biochemical networks that generate complex patterns of pleiotropy and correlations among traits. And when we think about complex phenotypes produced by metabolic pathways, what we can sort of imagine is that when metabolic pathways make more than one compound, it can also have more than one function. So if you imagine a toy single pathway that produces purple pigments in the flowers, but green defensive compounds, sort of as these stars are in the leaves, you might see some correlations between the two. But what's interesting is that the same compounds, let's say this gray star, my little, lost my pointer there, can have multiple roles. So the same pigment could be a defensive compound in a leaf, but also like a UV pigment in the flower. So just to sort of describe a lot, that metabolic pathways can be quite complex. My favorite pathway to think about is the flavonoid pathway. So don't worry too much about the details right here yet. So the flavonoid pathway is a well-conserved metabolic pathway with known structural genes. It's conserved in the plant kingdom and is responsible for several important ecological traits in plant life. This includes floral color, herbivore deterrence, antifungals, antibiotics, UV radiation protection, antioxidants, sugar transporters, nodulation, pollen development, a lot of very important features in a plant's life. And so my favorite part of the pathway is that of floral color. So this is generally made by the anthocyanin part, the specific branch of the pathway. So why is floral color interesting? I'm sure a lot of us are interested in floral color, so this isn't news to a lot of people, but just to sort of reiterate that floral color is a phenotypic trait that varies at all levels. So it varies within populations, between populations, and between species. It's important for both biotic and abiotic interactions, such as pollination and then say thermoregulation. And it's also a major phenotype in plant speciation. So throughout the course of my short scientific career so far, I've really focused on floral color and specifically what the role of pigments are in both micro and macroevolutionary processes. So that means things like local adaptation all the way through to speciation. And so like I mentioned earlier, today I'll be talking about two independent regains of floral color in the genus Petunia. And as I also said, this is my postdoctoral work that I've just finished with Chris Kuhlmeier. And so we can't bring up Petunia without thinking about pollination syndrome. So just to really quickly go through what at least my definition of the pollination syndrome is. So as we know, much of floral diversity is due to close associations with pollinator groups. And the combination of traits that attract, reward, and enable efficient pollination can sort of be grouped by particular pollinators. And those are called pollination syndromes. And so looking at the genus Petunia, we know that it's a young-ish genus in the Solanaceae. And in Petunia, the main isolating mechanisms between species are prezygotic, where you have both geographical isolation, and then when species are in sympatry, there's strong pollinator preference. There are three main pollination syndromes in Petunia. We have the hummingbird syndrome, as seen here, with characterized by red color, UV reflecting petals, and no scent. We have the hawk moth syndrome, where we have white petals, no visible color, UV absorbent petals, and very strong scent, usually at night. And then we have bee syndrome, which you see here. It's a blue-purple color. There's often UV patterning and some scent volatiles as well, generally during the day. Bee syndrome can be broken down into many different bee syndromes, but we'll just leave it as bee for today. So bee syndrome is actually the most common syndrome in Petunia, if we're just counting the species. And it's largely housed in one clade of Petunia, which is called the short tube clade. And all of these species look quite similar, other than size. But the group of species that we work with in the Kuhlmeier group is actually the long tube clade, which is much smaller. It's technically only 3 species. But in this clade, this has undergone several shifts in both trait combinations, floral trait combinations that is, and pollinators over a very short period of time. What's interesting is that morphology and geographic ranges are shared in this long tube clade, but the pollination syndromes vary quite a bit. So as you can see here, the main 3 species are Petunia axillaris, Petunia secreta, that's my laser pointer here. So Petunia axillaris here, Petunia secreta, and Petunia exserta. And then we tend to use Petunia inflata as sort of an outgroup that represents the short tube clade. Okay. All right, so thinking about shifts in pollinators, it follows that huge shifts in pollinators and traits requires changes morphologically, and it can be quite intense. But we know that all of these individual traits that make up the pollination syndrome are likely to be encoded by multiple genes. And in the Kuhlmeier group, we've been focusing on characterizing the molecular genetics of the pollination syndrome evolution and speciation by observing floral color, floral scent, and reproductive morphology. And my particular focus in the project was that of floral color. So in today's talk, I'll be focusing on these 2 sister species here, Petunia secreta and Petunia exserta. The 2 species have undergone some very interesting changes to pollination syndrome traits, and are a really interesting pair when some traits are shared. You can see a lot of the morphology is shared, but not all, while others like floral color, as well as reproductive organ morphology, are quite different. We estimate that the common ancestor to the long tube clade was likely colorless. More on this later. And so the 2 projects that I worked on were therefore to characterize the genetic mechanism of each of the colored species' regain of floral color. And so you can read more about each of these 2 stories specifically in these 2 publications, but I will do my best to describe them to you today. Okay, but first, a really quick note to the Solanaceae group about Petunia hybrida. So as I'm sure all of you know, Petunia hybrida has a very long history as a model species for scientific research. This includes decades-long research on floral pigments, And because of this, anthocyanin biosynthesis in particular remains one of the best known pathways of secondary metabolism in any plant species. And while the biosynthetic pathway is more or less intact in the natural Petunia species, so as we compare it from hybrida to the natural species, the transcriptional network controlling the pigmentation turns out can vary substantially in nature. Particular transcription factors and roles of said transcription factors can be quite different in the natural species. So while we are standing on the shoulders of giants when we look at genetics of the natural petunias, there's still quite a bit of work to do when studying them. So let's get started. The next thing I want to do is orient everyone to the flavonoid pathway. So many of you I know are quite familiar with it, but just to walk you through the pathway. So here's a depiction of the pathway as it exists in Petunia, where flavanols, which you can see on the top, and anthocyanins, which you can see on the bottom, are the major flavonoid compounds produced in the flowers. We have our two major transcription factors here, MYB-FL to activate flavanol biosynthesis, and flavanols serve as important UV copigments, and they also use the same core enzymes as the anthocyanins. The other main transcription factor is AN2, as you can see here, and this activates anthocyanin biosynthesis. Generally speaking, flux proceeds through the pathway like this. And at each of these sort of junctions here, these are called hydroxylating genes, where it actually adds a hydroxy group to the compound. At every one of these junctions, the precursor is either modified by FLS up here, that will make a flavanol, or DFR here, that will make it an anthocyanin. Generally speaking, the greater the number of hydroxy groups on the compound, the more blue-purple the anthocyanin is. Another thing to note is that Petunia DFR cannot accept this substrate, DHK, so it cannot make the true red pigment pelargonidin. And this will come up a little bit later. Okay, this is just to show you what these pigments look like when we visualize them. Okay, so a little bit more about AN2. So we know that the transcription factor AN2 is the main MYB transcription factor responsible for activating biosynthesis, anthocyanin biosynthesis in Petunia. And AN2 is additionally relevant in pollinator-mediated selection. We can observe in the field and we can demonstrate in the lab that having a functional AN2 is very important in making the shift towards bee preference in terms of pollination. And having a non-functional AN2 and losing that visible color is really important in shifting pollinator preference in terms of hawk moths. So it's relevant in terms of speciation is the point. And then it follows that sort of a similar story goes with MYB-FL with the flavanols. So having a functional or non-functional MYB-FL and therefore plus or minus flavanol pigments strongly influences hawk moth pollination. This is particularly important for the white flowered Petunia axillaris, which has strong UV absorption, which looks like a black flower using a UV camera. When you look like this, you have high amounts of flavanols and hawk moths really like it. When you don't, you look reflective and hawk moths generally don't like it as much. Most of the other Petunia species don't have very high concentrations of flavanols. So the first step in Petunia to achieving an intense visual anthocyanin pigmentation is actually to disable flavanol biosynthesis. If the Petunia axillaris allele of MYB-FL is present, then flavanols are synthesized in high amounts, leaving less substrate available for anthocyanin production. So it's therefore not surprising when we looked at these two colorful species that MYB-FL is deactivated in both of these species in slightly different ways. The other interesting thing is that AN2 is inactive in the long tube clade. So we predicted that AN2 would be responsible for anthocyanin activation in the long tube clade, as it is functional in Petunia hybrida and it is functional in the short tube clade, as you can see here in Petunia inflata. So here is a depiction of the gene. We have ATG and it just codes beautifully. However, when we sequenced AN2 across the whole long tube clade, we saw that this clade harbors several different haplotypes of inactive AN2, where we have a lot of different haplotypes with different early stop codon genes. Furthermore, we noticed that AN2 is definitely non-functional in the colorful Petunia exserta, which is puzzling. And I'm going to say, hold on this, and I'll tell you about it in a little bit. So certainly puzzling. So if AN2 is supposed to be the transcription factor that turns on anthocyanins, our question was, what turns on anthocyanins in Petunia secreta? So as we can see here, Petunia secreta does make anthocyanins, makes plenty. It is visibly purple, and we can detect a lot of anthocyanins in there. So if we know that AN2 is non-functional in Petunia axillaris and exserta, we were unsure about what we would find for Petunia secreta. However, we found that AN2 is indeed functional in Petunia secreta. But the interesting thing is that the AN2 haplotype, which you can see here is H11, this haplotype that Petunia secreta harbors seems to have been derived from a non-functional H10 haplotype. And this haplotype is found in both axillaris and exserta. What you can see is that there's a 2 base pair compensatory deletion, which restores the reading frame of AN2 in secreta. So while it's a slightly different amino acid sequence here, you can see it's just missing this K, essentially. It codes for a functional protein. So to demonstrate the functionality of the protein, just to quickly check, we overexpressed the H11 haplotype in the white Petunia axillaris. And you can see here that it restores floral color. Not quite to the same intensity, but it does restore that color. We also silenced this secreta AN2 in Petunia secreta to demonstrate that it was the transcription factor that is necessary for anthocyanin-based color. And here you can see white sectors. So then we wanted to know a little bit more about the history of this strange AN2 haplotype. So we performed an ancestral state reconstruction on the haplotypes, and we saw that the most likely scenario is that this functional H11 haplotype is phylogenetically most closely related, again, to the H10 non-functional haplotype. Interestingly, H11, the functional H11, is only found in Petunia secreta. We never found it in any axillaris or any exserta. Neither did we find it in any of the short tube species. So this further supports our claim that AN2 was subsequently and somewhat recently resurrected, if you will. All right. So then we looked at gene expression. So now we see we have a functional AN2. So what does it do? Does it sort of just restore gene expression, or does it do something else? So what you can see here is a lot of graphs, but what I want you to sort of focus on is this column A right here, where we have the dark purple bar representing Petunia inflata. So this is sort of representing the more ancestral and certainly the more common AN2 environment. And then we have the pink bar, which is Petunia secreta. And looking through all of the genes, you see very, very similar gene expression patterns between the 2 purple species. And we've stuck the white Petunia axillaris in for comparison. So the point being, high AN2 expression upregulates anthocyanin branch of the pathway in the purple species, in both of them. And subsequently, low MIB-FL expression, which does happen in Petunia secreta, decreases that flux towards flavanols. So we see high anthocyanins, low flavanols. So more or less a restoration of anthocyanin expression patterns. In terms of the chemistry, the Petunia secreta AN2 does, as we can see, restore visible anthocyanins. And what we see is a similar, if not the same in concentration, but we see a similar composition where we have a lot of malvidin and petunidin and a little bit of delphinidin. And the same goes for Petunia inflata here. So similar composition to make that purple color. So to summarize this part of the story, the resurrection of AN2 through a frame restoring mutation in Petunia secreta does seem to restore visible color. It is not simply a reversion or a retention of the ancestral allele. And together with a graduate student in the lab, Martina Luthi, we've also demonstrated, that, it's not gonna let me do it. We've also demonstrated that Petunia secreta AN2 restores bee pollination. Oh gosh. I hope, does that look weird on your screens or is that just mine? Okay, well, is it bad on your screen, Chelsea?
Yeah, it's like black. I don't know if there's like white parts that are missing.
Okay. Well, there's a graph, and we did some pollinator assays. Sorry for that. And you can see that AN2 does restore bee pollination, and it decreases hawk moth pollination. Okay, clearly, thank you. All right, so clearly it wants me to stop talking about Petunia secreta, so I will. So now we've sort of described Petunia secreta, and it's sort of like, in a way, a simple mechanism. Now we focused on the mechanism of the transition to red color in Petunia. So red color is really interesting to a lot of people. The evolution of red flower color has often been associated with the transitions to hummingbird pollination. Not always, but often. And there are two major biochemical pathways with which to make red floral color. You can make them with carotenoids, anthocyanins, or with both. Petunia, however, only uses anthocyanins for its main floral color. And it can be useful to think about the evolution of red in the context of floral color hue transitions, with red flowers most often having transitioned from a blue or a purple state. There are also a few very well-known cases describing the mechanism of the transition in several systems, as you can see here. One of which is in the Solanaceae by Stacy and Iachroma. So transitions from blue to red is usually the direction in which red arises. However, with Petunia, it's a little bit less clear what the direction was. However, we are fairly certain that the common ancestor to the long tube clade here, since we know that it had a non-functional AN2 and it probably had a functional MYB it just seems likely that this flower was probably white. So in which case, it's still harder to say if it was a white flower and then it picked up pigmentation first and then went red. Really hard to tell. So we sort of had to take the idea of both gaining color and a possible transition in thinking about Petunia exserta's red color. Almost all the other petunias are purple. Suddenly we have red. So I mentioned before that AN2 is broken. So it really left us wondering how Petunia exserta can actually make anthocyanins. And then additionally, why is it red and not purple? So the first thing we did is we did a handful of QTL experiments just to see what, visually, what is the QTL sort of environment? What's the genetic basis for anthocyanin content in these species? So knowing AN2 is not responsible, we can see on the right-hand panel, this is an Oxalaris by secreta cross, and chromosome 6 is where AN2 lives. So here we know AN2 is functional, we see a huge QTL for there. On the left-hand side, this is Oxalaris by exserta. We see no action on chromosome 6. We know AN2 is non-functional, that's great. The point being, there is a very different genetic mechanism behind anthocyanin content in exserta compared to secreta. So not the same genetic mechanism. So therefore, we basically have two questions. What are the biosynthetic gene expression changes that allow for the red versus purple color in exserta? And the second is, how did exserta regain anthocyanin biosynthesis? Specifically, what is the transcription factor that replaces AN2? So since chemistry is one of my favorite things to do, we just did a full panel on what are the anthocyanins in our long tube clade species, and then we still retain Petunia and Fla to to show the other group of pigments. So focusing in on exserta here, we see that it is different in its composition of anthocyanins. It is unique because we see both cyanidin and delphinidin as the main anthocyanin pigments. We do not see pelargonidin, which is the most common red pigment species. So in addition to having this unique pigment composition compared to the purple species, we also have an odd concoction to make the red color. So using cyanidin is not uncommon for red, but certainly using delphinidin, which is typically a purple pigment, is much, much less common. The other part of, of the anthocyanin that I'm not going to show you, but I'll just tell you, is that when we looked at the specific types of anthocyanins, we noticed a unique modification pattern in that there are no acyl groups attached to these anthocyanins. And that'll come up again a little bit later, but just know these particular subgroups on the anthocyanins were missing in the red species compared to the purple species. So visually, where does that put Petunia exserta in the pathway? So I've just sort of put the flower here to show you where most of the pigments are. So going back to that flavonoid pathway, we looked at the coding sequences next of all of the biosynthetic genes in the pathway, and we compared Petunia exserta coding sequence to the sister species. We found no major or obvious mutations in the biosynthetic genes. So we focused our attention instead to changes in gene expression. So what about predictions there? So in order to be able to produce cyanidin compounds in Petunia exserta, we expected higher expression of this gene right here, F3'H. We would also still expect some expression in F3'5'H due to the delphinidin present. So the other thing that I mentioned previously is that we observed no acylated anthocyanins, but we also observed very few of the methylated anthocyanins, which are down here. So those are peonidin, petunidin, and malvidin. So I've sort of just boxed this area out where we'd expect either low expression or something weird. So those were sort of our expectations. So in tandem with qPCR and RNA-seq, we used virus-induced gene silencing to determine which of these biosynthetic enzymes are necessary for the red phenotype. So I'm just going to show you the results from the VIGs, but the qPCR and gene expression things are also in the paper if you'd like to see that in more detail. So walking you through this figure here to the right, we have the control in the left-hand column for each of the 3 colorful species. And then each of these columns is how we silenced one gene at a time. And so what you can see here is that in exserta, you see for both F3'H and F3'5'H that you see infected sectors here. The important part that suggests that both these enzymes are necessary is that we only see light infected sectors rather than white. So that means that both cyanidin, as controlled by F3'H, and delphinidin from F3'5'H are both necessary to make that full red color. Now going down here, I'm going to get back at that acylated information. So AAT, which you can see here where I've put the little lightning marks, is the gene responsible for acylating anthocyanins. And what you can see is that when we silence AAT in exserta, nothing happens. And that makes sense because, as it turns out, that gene is extremely lowly expressed in exserta. It is highly expressed in the purple species, which make acylated anthocyanins. So you can see that silencing this gene, again, has no effect on exserta, but what it does to the 2 purple species is it makes them pink. So it's actively shifting the visual color from something blue-purple to something more pink. And so it's sort of like a roundabout way of saying AAT is lowly expressed in exserta, and that's allowing it to stay red instead of shifting to a more purple-blue sort of color. The other thing that's important to know about AAT is that it's sort of a gatekeeper enzyme to get to these methylated anthocyanins. So downregulation of AAT is actually quite important in that it makes It allows for those exserta red anthocyanins to stay red, and it also prevents those methylated anthocyanins, which tend to be more purple-blue, to be made in any large amount. So, okay, what turns on now anthocyanin biosynthesis? Anthocyanin biosynthesis is turned on by, generally speaking, a triplex of proteins where you have a MYB transcription factor and then two partners. One is a BHLH and the other is a WD40. In Petunia secreta and inflata, we know we have AN2, and this is the MYB transcription factor that activates biosynthesis. So we therefore set out to figure out what is the MYB in Petunia exserta. The other two partners were completely intact and quite normal. So the first thing we did is we mined the Petunia genomes that we have. So in the Kuhlmeier group, we have two excellent genomes of Petunia axillaris and Petunia exserta. And then for this paper, I also generated a genome of Petunia secreta. So we mined these three genomes for all of the MYBs in those three species. And I'm showing you here on the left, just a depiction of the subgroups of MYBs that are associated with flavonoid pigmentation. The point is that no new MYBs were found in any of these pigmentation related subgroups. So no like new genes were discovered. So that led us to believe that there must be some sort of functional shift in a current MYB. So with that, we conducted an RNA-seq and allele-specific expression experiment with all three species. And so this is sort of a depiction of focusing in on subgroup 6 MYBs, which are the anthocyanin activators. And this is just to show you we had 3 separate experiments with each of the colorful species compared to the white axillaris. But I'm going to actually shift the view to this, where I've converted those raw gene counts to log 2-fold change. So what you see here on the left is a value of 0 is sort of the white axillaris, and then you can see higher expression being above 0 and lower expression being below 0. 2 candidates popped out immediately that you could see that these 2 genes, deep purple and PH4, are highly expressed in Petunia exserta, especially compared to the other 2 colorful species. So deep purple is abbreviated as DPL, and PH4 is actually not a MYB subgroup 6. So PH4 is in its own weirdly annotated subgroup, and PH4 actually activates vacuolar acidification. And vacuolar acidification is also incredibly important in color hue, essentially. So this was interesting, a little surprising, and also interesting. The other interesting thing about deep purple is that it is a MYB subgroup 6 anthocyanin activator. What is also interesting is that deep purple in Petunia hybrida is known to activate the flower vein pigmentation. So not the petal pigmentation as such as AN2, but the veins. So interesting. The other curious thing about deep purple is that if you look at the QTL map, we have a lot of QTLs, but deep purple is located on chromosome 7, which is in fact a minor QTL. So deep purple is actually quite compelling to be the possible AN2 replacement. So I'm going to come back to PH4, but now we're going to shift our focus to deep purple. So what we did is the same sort of approach as with the biosynthetic genes. So the first thing we did was we're like, great, let's silence deep purple to see what happens. So we silenced deep purple in all 4 of our species to determine whether deep purple is necessary for anthocyanin biosynthesis. And indeed, what you can see is that when we silence deep purple in Petunia exserta, you get white anthocyaninless sectors. And you can see the chemistry here, where you see a strong reduction in anthocyanins. It's not perfect silencing and perfect, like a 0 amount of anthocyanins, because of the nature of how virus-induced gene silencing works. However, you can see visually that those are white sectors, and it is also unique to Petunia exserta. So deep purple is responsible for anthocyanin biosynthesis in Petunia exserta only. So this suggests that the upregulation of deep purple in Petunia exserta is responsible for anthocyanins. So the next step we took was to demonstrate that deep purple can actually activate anthocyanin biosynthesis. So what we did is we overexpressed deep purple in the white Petunia axillaris. And what you can see here is that deep purple does indeed induce anthocyanin biosynthesis. In particular, you can see this in the purple color of the transgenic plant. And you can see on the right, the shift in the chemistry where we see a higher accumulation of delphinidin. You can also see from the bottom panels where I have gene expression here, that in the purple boxes, which is the overexpression line, deep purple clearly activates anthocyanin specific gene expression from DFR downwards, where you can see high expression of all of the anthocyanin specific genes. It does not seem to differentially activate the hydroxylation genes, which would therefore control the hue of the color. So what we can say is that deep purple is the on-off switch for anthocyanins. One of the most obvious things you might be thinking right now is, why isn't the flower red when you overexpress this gene? And we also wondered that, but then we just sort of sat down and like, okay, let's think about this. This has to do with the specific changes in expression of the biosynthetic genes that I mentioned earlier. So the biggest difference between Petunia axillaris and Petunia exserta is that Petunia axillaris produces tons of quercetin. And you can see that here as these big gray bars. This is so much quercetin that you can very easily break a UPLC machine, especially when the machine is not yours. A lot of quercetin. And because of that, DHQ, which is the precursor shared by quercetin as well as cyanidin production, if all of the DHQ is being made into quercetin because of a functional MIB-FL, you're just not going to get cyanidin. So DHQ in Petunia axillaris, even though we're overexpressing deep purple, is still consumed by the FLS enzyme. So what we're left with is activity by the two copies of F3'5'H in Petunia, one of which seems to be better at synthesizing anthocyanins from another precursor, DHK. And thus we only get the delphinidin purple color and lots of flavanols, rather than a mixture of cyanidin and delphinidin red. I'm also happy to talk about that further later. But anyways, the point is, when we overexpress deep purple, we regain anthocyanin biosynthesis. Now, getting back to pH4, this one was a little bit surprising in terms of our results. So as we know from really lovely work from Francesca Quattrocchio, as well as Ronald, that pH4 is a MIB transcription factor, and silencing pH4 in Petunia hybrida, as you can see here on the left, it changes the vacuolar pH in the petals, and that leads to a shift in hue. And as you can see here, this is what it looks like in Petunia hybrida. And when we silenced pH4 in Petunia inflata, we see a really similar phenotype where you can see dark blue sectors. So the sectors end up going more basic when you silence pH4. So this is wonderful. We thought, great, we know exactly what to expect for the long tube clade. False. Instead, what we saw was that when we silenced pH4 in the long tube clade, and especially in the colorful species, this induced anthocyanin loss in both Petunia exserta and Petunia secreta. It's important to note that this phenotype is not specific to Petunia exserta, but is shared by sister Petunia secreta. So therefore, while pH4 is clearly important, it is not the actual candidate gene for anthocyanin biosynthesis that's unique to Petunia exserta. I followed this up with experiments in measuring petal pH in these two species, and we could not actually detect a difference in the virus-induced gene silencing individuals in petal pH. So we were a little bit puzzled by this. So what is pH4 doing in the long tube clade? we think that in Petunia inflata, it is doing the thing that has been demonstrated in Petunia hybrida, where it's actually changing the vacuolar pH, but we don't have evidence for vacuolar pH differences in the long tube clade. So our best guess is to look for what pH4, what it's doing in Petunia hybrida. So pH4 controls a series of proton pumps in the vacuolar membrane that controls the pH. And given that information, we think that the function in the long tube clade is just simply different. We see a loss of color, complete loss of color, rather than a change in the pH and a change in the anthocyanins. So our best guess is that pH4 must play an important role in the transportation of anthocyanins into the vacuole. That's to be determined. But the point is that whatever pH4 does, it doesn't affect Petunia exserta exclusively, but it is also clearly necessary for appropriate anthocyanin accumulation. And this is just to show the petal pH. Okay, so concluding remarks about Petunia exserta. What is the genetic mechanism of the shift to red in Petunia exserta? So first, predicted expression changes in the hydroxylating loci, genes rather, allow for the production of cyanidin and delphinidin, which together allow for a redder hue, more than purple. Second, the downregulation of anthocyanin modifying enzyme AAT prevents anthocyanins from becoming very dark or intensely purple. And last, we observed upregulation of 2 MYB transcription factors and demonstrated their modified roles in Petunia exserta. Deep purple shifts from vein pigmentation function in the other sister species to replacing the role of AN2 in the petal limbs for anthocyanin biosynthesis activation. pH4 does not appear to regulate vacuolar pH in Petunia exserta in the same way as it does in the short tube clade, but it still has a necessary role in floral color phenotype. And so, like I said, if you'd like to read any more about these stories, here are the 2 publications, and I'd be happy to answer questions about them. And just as the last comment, this is what I'm doing now. I have left the Solanaceae and I've returned to my original love, which is the genus Silene in the Caryophyllaceae. And I am currently working at the Harvard University Herbaria on the speciation processes and evolution of floral color in North American Silene polyploids. So with that, I'll take any questions if there's remaining time, and thank you for your attention.
Awesome, thank you so much, Andrea. So if people have questions, you can just write that you have a question, or you can write it out and I can read it out for you if you want. Let's give people a second. It was an awesome talk, Andrea. It's so thorough, and I don't know, pathways are always intimidating, so it's always so great to hear. Cool.
Okay, I see comments of it being weird in the text. Great, love it.
Okay, so Stacy has a question. So my video is being weird, but I was wondering why Andrea thinks MIBs can switch jobs like this.
Oh, that's a great question. We thought really hard about this, and I think there's no simple answer, but I think given the propensity of MIBs to easily diversify, I've always thought that because they can diversify their phenotypes over evolutionary time, I think it's just quite easy to regulate different ones at different times with just a simple switch. So that doesn't make sense. I think that if you have a MIB that can already do one function, then it's easier to shift into another function, especially when it's within the same tissue and relative developmental time frame. So with Deep Purple and AN2, they have really similar, not in exserta, but in like Petunia hybrida, for example, they have really similar expression patterns in terms of the bud development as well. So I think if it's already doing one thing that's quite similar, it's relatively easy to be able to shift a tissue specificity. But it's a really, yeah, I don't know if I have a great answer other than if it's already doing something similar, it might be able to just neo-functionalize essentially.
Yeah, she responded with a good point. As long as MIBs might be
Oh,
active, they might be around to co-opt. Thanks.
yeah,
Luke
yeah.
has a question. Did you want to read your question, Luke?
Sure. Yeah, I was wondering if you saw any evidence of if there are changes in the DNA binding domain in pH4 and P
Mhm.
exerta, or if you think it's more
Yeah,
likely, could it be a cis regulatory change on some of the targets?
yeah. Really great question. There are no obvious changes in any of the DNA binding sites of pH4, which is both frustrating and interesting. And when we did the allele specific expression, which I didn't talk about in this talk just due to time, there's clearly it's a cis regulatory situation. So both deep purple and pH4 had very intermediate values of allele specific expression, which would suggest both cis regulatory and trans regulatory actually. Our best guess for pH4 when we looked at the promoters was that there's very obvious cis regulatory changes, but to test that in practice can be really quite difficult. And it would be great if somebody did that, but then I left. So yeah, it's a wonderful question. And the answer is like mostly cis, a little bit of trans. We did spend some time looking to see if we could find the trans factor that maybe deep purple and pH4 would share, but also the trans factor that deep purple and AN2 would share. And we just, we did actually a lot of fine mapping with the QTLs, and because of the strong influence of NIB-FL, huge pleiotropic effects, it was actually quite difficult to finish that fine mapping. So hopefully somebody can figure it out and find that mysterious trans factor, which we've labeled as a mysterious ANX. But yeah, great question. I would love to know the actual answer as well.
Awesome. Is there any more questions for Andrea? You got a lot of congratulations. You'll be missing Solanaceae.
I'll miss Solanaceae too.
That's great. All right. Well, if there's no more questions, we can wrap up for today. Join us next week for Jay Goldberg. He's going to be talking on a sticky situation, the mechanisms underlining the natural trichomes dimorphism in Datura wrightii. So thank you so much, Andrea. See you guys next week.
Thanks for having me.